There are 2-3 people still have not joined. You send a message to them that they have not joined. And I think Laksangi, we can start, because he has also to leave. Quickly,
Okay,
we
sir.
can start.
Okay. Yeah. Good evening, one and all. Today we have Professor Sanjeeva Srivastava as resource person for the second session of this RC course. I am Dr. Lakshangi and I am privileged to introduce him. Professor Sanjeeva Srivastava is a dynamic personality and a proactive researcher. He is the head of the Proteomics facility and a professor at the Indian Institute of Technology, Bombay. Currently, he is appointed as a full-time visiting professor at Department of Laboratory Medicine, School of Medicine at the University of California, and visiting professor at various universities abroad. Sir has obtained his PhD degree from University of Alberta in 2006, postdoc from Harvard Medical School 2009, and then he started his journey at IIT Bombay as professor. Sir has established one of the best state-of-the-art proteomics facility in India. Sir has over 150 publications in reputed national and international journals. He also has more than 220 patents to his credit. Sir is recipient of various national and international awards. Some of them to name are Young Scientist Footstep Award Canada, the Martha Piper Award for Communications Excellence, the Young Scientist Award from the Department of Atomic Energy and Department of Science Technology India, IIT Bombay's Young Faculty Award, and so on. He is Fellow of the Royal Society of Biology, Royal Society of Chemistry, and Royal Society of Medicine, and serves various organizations such as HUPO, that is the Council of Human Proteome Organization. Recently, he has started the Cancer Moonshot India program to accelerate cancer proteogenomics research, and India became part of the International Cancer Proteogenome Consortium. Sir is also a scientific filmmaker, and his documents have witnessed various national and international film festivals. Sir plays a major role in proteomics education, training, and outreach, and has emerged as a leader and catalyst of the proteomics research and education globally. With this brief, I will really say it is brief introduction. I welcome Professor Sanjeeva Srivastava and I request him to deliver his lecture. So, welcome.
All right. Thank you very much for the very kind introduction. And thank you, Dr. Sandeep, for this initiative of annually you are conducting this program of UGC faculty training program, which is, I think, quite good initiative and important. And that's why we're always there to help out in these sessions. So if you allow me to share my screen, I can start my presentation. See my screen now? Was my screen visible?
No.
No?
No, sir.
I'm just trying again. Okay, some bandwidth issue. I'm just turning off my phone and turning off my video. Let's see if this works. Please let me know if it is working now.
Yes, sir. Yes, sir. It is fine now.
All right.
Be a muscle. Make a bone. Carry nerve signals. DNA tells our cells exactly what to do and when to do it. That's why DNA is often called the blueprint for life. But how do our genes give such orders? Each gene consists of a recipe for a certain protein. Proteins are the machines and workhorses of our body. Proteins trigger cells to perform specific tasks, enabling the cells in your eyes to function differently from the cells in your heart or brain.
Our bodies are made up of about 100 trillion cells, which each contain thousands of different proteins. The collection of proteins within a cell is called the proteome. The proteome is the counterpart of the genome. Our DNA remains the same throughout our lives, but our proteome changes. This explains how the grown-up flying butterfly can become something so distinct from the young crawling caterpillar it once was.
Few proteins work alone. They interact and form relationships with each other, and the nature of these interactions changes over time. You could say proteins have their own social network, just like us. Some proteins have lots of friends and contacts, while other proteins only have a few. Sometimes, a protein will break up an interaction and start looking for a new partner.
With approximately 10 billion protein molecules, one single cell has an interactive network that is as complex as that of all humans currently living on planet Earth. These protein networks determine the health of a cell, and thereby the health of the entire organism they are a part of. So it is easy to understand that the study of these proteins and their relationships is crucial. This study is called proteomics.
Understanding how cells work has made it possible to develop revolutionary drugs that are more effective and have fewer side effects. To detect and identify different kinds of proteins, we measure their unique fingerprints with a technology called mass spectrometry. This technique can determine the identity and, indirectly, the function of proteins.
The process involves shooting charged protein molecules or fragments thereof into a vacuum tube and measuring how long it takes for them to fly to the other end. This flight time depends directly on their mass. Another way we learn about the molecules is by making them collide with gas molecules, which causes them to fragment into smaller species. By measuring the mass of the fragments, we can determine what the protein's building blocks are and can thereby identify it. In one single experiment, we do this for tens of thousands of proteins and roughly 1 million fragments. This meter shows the huge amount of data we are constantly producing by measuring proteins. Gigabyte after gigabyte, this information helps us to precisely identify proteins and the social networks in which they exist.
But proteins are not unique to humans. Every living species on Earth is built up of proteins. Jellyfish, polar bears, tulips, and even bacteria. For this reason, study of the proteome will ultimately lead to better knowledge, not only of diseases and the effectiveness of drugs, but also of how plants can adapt to dry or salty environments, and how we might eventually be able to combat bacterial resistance. Excitingly, in recent years, scientists have learned how to reprogram the proteome of skin cells in a lab and turn them into stem cells. In other words, scientists are now able to tell cells what to do. For instance, regenerate tissue or repair a damaged organ. New therapies for a healthier society are on the horizon due to the fascinating research of proteomics.
All right, so welcome to Proteomics, and I'm Sanjeev Srivastava from IIT Bombay. I will give you in next 1.5 hour time some of the proteomic technologies overview, how they work, what are the concepts involved, and then I'll focus mainly on the mass spectrometry-based proteomics and some of the applications which we are doing in my lab here at IIT Bombay. So, if some of you have already visited our campus, you might know it already, but some of you have not. Please do visit. It's a beautiful campus. It's one of the institution of eminence, and we'll be doing some workshop on advanced proteomic technologies in February next year. So again, that could be an opportunity for you to come and look at the campus. I'm in this department of biosciences and bioengineering, which is primarily focusing on postgrad education. Large number of MSc, MTech programs we have, plus PhDs are the main research workhorse. 250 plus students who are doing research in different laboratories. Before I start, just a disclaimer that we have this startup company from my lab, Proteomica International Private Limited, to provide proteomics and omics-based solutions. This company is incubated at Sign, but there is no conflict of data. What we will be talking today with this company activity from Proteomica. So broadly, my lab has been working on a couple of projects. One has been on technology or platform development, assay developments. Essentially, many engineering undergrads and technology-related people we get, and always aim is how to make the best use of different type of proteomics instrumentation, new software developments, new type of assay or test development. On the biology side, we look at the brain tumors, cancer, and also some work we have done on the infectious disease like malaria and recently COVID-19. Many collaborators come, they want to learn proteomics on their own biological samples or they want to perform proteomics on the model organisms. So we also end up doing proteomics of other organisms, but that's not our focus of research. So I'll first talk to you about what is proteomics and what kind of proteomic technologies are there. I'll give you some concept of mass spectrometry and some of the non-mass spectrometry technologies as well, including protein microarrays and label-free biosensors. I'll take few examples of what we are doing on using proteomics, especially I talk about COVID-19 and some of the more recent work on the brain prot. So I'm sure you're aware of, first of all, the genomics, which really laid the foundation of all the other technologies and gave us that now we have ability to look at the blueprint of life, all the genes of human. Following that, idea was, can we also look at all the proteins which are expressed by the genome? And if we have 20,400 or so genes in human, uh, that many proteins should be present. And can we at least have the experimental evidence of all of those proteins, uh, which we can try to understand? So that's one of the major goal of field of proteomics, to study the proteins, their properties, and understand the integrated cellular processes, how they are being governed using these proteome. Many important processes like protein modifications, protein localization, protein-protein interactions, which are shown on the screen, are very important in this process. Just I want to draw the some comparison from genomics versus proteomics. Again, like many people who work in this area of genomics, genomics is very powerful technology. However, from these examples, what it will be apparent that the genome remain same or consistent throughout the developmental stages of these model organisms like butterfly or frog, which I have shown here. While as the organism grows from larva to pupa, adult form, and the adult butterfly, then what we see, the proteins get different, and proteome is very dynamic. It actually provides the ability for these organisms to manifest different type of coloration, different type of shape, different ability to cope up with the environment. And that's really apparent in case of frog when we have the stack code and the adult frog. Both are in very different environment from the aquatic to terrestrial. And again, the genome remains same. What makes them different is the proteome, which gives them the capability of performing these functions. So I hope that is now clear that both of technologies are very important. But while genomics provide us more of the blueprint information, proteomics gives us more dynamic and the functional information. I'm sure here many faculty members who are joining this course, uh, your own laboratories are working on several projects, and irrespective of whether you do proteomics or not, but at some stage you will be doing or you must be doing protein extraction for different projects. And so if you have a good biochemistry lab, or if you can be very creative and intellect about looking for good protein extraction methodologies from given sample, I think you have already done 50% of proteomics because the rest of the things are built on the technologies. But input samples become very crucial, and that's where extracting protein for the proteome scale analysis is quite challenging. And many times goals are to look at the whole organism, for example, whole of the yeast, whole bacteria, whole of the human. But then most of the time that's very complex task. Then people look at some specific organs, especially in the human case, or look for specific organelle, sometimes specific cells. So many of these things,
Arushi.
depending upon what is the your context of proteomic analysis, a different type of extraction methodology could be planned accordingly. So this is first part, which is essentially there are many methods in literature, like you can do TC acetone based precipitation, you can do acetone alone, you can do triazole based extraction method, you can do urea lysis method. Many methods are there, but what works on plants may not work on fungus, may not work on bacteria. What works on bacteria may not work on human sample. What one works on the one type of organelle may not work on the membrane protein. So again, as I said, your intellectual knowledge about your sample type and your understanding of biochemistry for your given sample is very crucial to get started in the proteomics. But I'm sure, as I said, all of you must be doing it already in your lab. So essentially, you're already pretty much ready with the first step of protein extraction for the proteomic analysis. Now, since we are talking, when you've extracted the protein in a given test tube, what we are talking, maybe in that given test tube, you have 5,000 proteins, if you're looking at all the yeast proteins. So do we have ability to now identify all of those 5,000 proteins? For doing so, you want to first separate the complex protein mixture into smaller, simpler fractions so that you can get better proteome coverage. And that you can do either gel-based approaches. Sometimes people run their complex sample on LDS page, chop down certain parts of the gel pieces, and then perform the in-gel digestion, followed by mass spec analysis. Sometimes gel-free methods are being used. Different type of chromatographic methods are being used to fractionate your samples. Then finally, idea is, can we now run them in the mass spectrometers? Different type of mass specs are available, whether it's multi-TOF based or ESIQ TOF based. And then you perform database search from different type of bioinformatics workflow, and see now you are able to identify thousands of proteins from the given sample. Once you have performed this kind of workflow, and let's say at the end of it, if you are doing the differential proteomics, for example, you are looking at control versus treatment comparison, so then you want to now characterize those proteins for some of the good protein which you have identified for which there is no function assigned. So you'd like to characterize them. And that's there are many type of functional proteomics technologies like protein microarrays, label-free biosensor like surface plasmon resonance, as well as some of the structural technologies, X-ray crystallography, and of course, protein purification followed by looking at different type of electron microscopy. So many of these things will be utilized to do the characterization of the protein, which is essentially some of the chemistry labs are already doing these kind of structural work. But new proteomic technologies like microarrays and label-free will also be helpful for you to assign the function. So I hope you are clear now that we are talking about four major steps. But at each step, you have many technologies available, and you have to really choose which one will work best for your objective and your given sample type if you want to do the proteomic analysis. So I'm hoping at the end of my today's session, you'll be better informed or more informed to choose which methodology, which techniques will be best suited for your sample type. What should be, what kind of workflow you can make. While of course I cannot provide you the protocol right now in terms of, because that cannot be one generic protocol, but you can at least come up with the workflow for your project, what is of your interest. Let me first take you to the very basic proteomic technology, which are gel-based proteomics. When I say gel-based proteomics, I'm sure everybody here would have run the SDS page gel to separate protein in one dimension. But if the proteins are, we are talking about in thousands of number in a given sample type, people also want to separate those in the two-dimensional electrophoresis. So what you see, this kind of cartoon image is two-dimensional electrophoresis to have the two different dimensional separation of the proteins. These are the 3D views of those protein spots. And if you want to become more quantitative, you can tag your control and treatment sample with different type of fluorophores, Psi3, Psi5, for example, and you can now look at the patterns of those images on the 2D dye images. So these are the kind of various technology which I'll be talking in next couple of slides before I move to the major workhorse, which is mass spectrometry-based technology. The two-dimensional electrophoresis looks for two different dimensions of the protein separation. Isoelectric focusing is the first dimension separation based on the PI values. And then the second dimension looks for the molecular weight-based separation. So you're trying to utilize two different properties of the protein and trying to separate them on the gel so that you get the spots like pattern. And those spot denotes what is the pi value and what is the molecular weight of a given protein, right? So I'm sure some of these basic concepts you might already have. Let me show you one short video of the DICE technology, difference in electrophoresis. And as I will be showing you, I'll keep explaining the concept of it as well. So, moving from 2D, now when we are talking about, let's say, quantitative proteomics using gels. So you have one sample which is control, other sample which is treatment, right? And let's think about, we have three controls and three treatments. All the three control sample you have labeled with one dye, Psi3 dyes, and the treatment sample you have labeled with Psi5 dyes. Additionally, you are also trying to make a pool of these samples. These six samples, you are trying to make a pool. For example, here you see uh, the first sample, let's say we are labeling control sample with the Psi5, and treatment samples we are labeling with Psi3. Of course, this can be your choice. You can do dye swapping. Additionally, you are taking all the six samples, a small aliquot, and pooling them in one common tube and labeling them with third dye, which is Psi2 dye. So now what I showed you was two samples, but you made three tubes and labeled with Psi5, Psi3, and Psi2, and you have mixed them all together in one tube, which now you are separating them in the first dimension separation based on the pi value, and then followed by second dimension separation based on the molecular weight. The 2D concept, only difference was the sample labeling. But now, since you have labeled the sample, you're running them on one gel only. You have both control and treatment. So C1, T1 will be running on one gel, right? So you generate two images of that, and then you also have internal standard image just from Psi2. So now you got three images from one gel. And likewise, you got C2, T2, an internal standard image, and C3, T3, an internal standard image, right? So I hope you're getting clear that we got three control, three treatment samples. And after doing the dye labeling, we got three images from each one of those. Now this is standard or internal standard sample image that will be same for all the three comparisons which we are making, because we have made a pool of it from all our samples, right? So I told you to take pool of all six samples so that whatever variability we have of all the proteins from each population, that will be accounted for in this sample. So this, now we got three comparisons and three images of internal standard. These three internal standard images will help us to make our analysis very automated. Now we are looking at biological variation analysis, or BVA module, where we are comparing our control with treatment, and what is helping us to align the gels is internal standard. The internal standard in all the three comparisons which we are showing remains same, right? Because that was the blueprint of all the proteins which we pooled, and that is helping us for the internal standard normalization. Now, this is very important concept which is known as co-detection. And once we have done the alignment of these gels using the internal standards, now we can have n number of comparisons to be made. So otherwise, you think about running too many gels will have a lot of variability. Gels running artifacts and different reaching differently, many problems. At the end of the analysis, you would like to see like which proteins are differentially expressed, what are the fold changes, which type of biological pathways they belong to, which type of heat maps you can assign them, looking at their patterns of control and treatment, differential expression analysis, PCA you can perform, pattern analysis you can perform, heat map. So many things which you can do, which will be helpful for you to understand the biology of it. I hope in a very quick nutshell you were able to get some idea for the 2D gel or gel-based proteomics. I wanted to spend less time on it so that I have more time to talk, move on to mass spectrometry-based proteomics. But nevertheless, gel-based technologies are fundamental technologies, and they're important for getting started in proteomics. If there is any question in this part so far, please ask me questions. I can stop for a minute. I'm hoping all of you are taking this course because you feel value to take some of these concepts back to your labs, right? So please do make it more interactive. I am not giving this lecture like a conference audience, or not like my class students, but rather to our colleagues who will be able to take some of these things forward in their labs. So make it more interactive. Please feel free to ask questions wherever something is not clear to you, because otherwise I have a lot of things to talk and I'll keep moving forward. But feel free to interrupt me and ask me questions after each segment when I ask you. Yeah? All right. So let me move on to next part, which is mass spectrometry based proteomics. Uh, This is one of the very growing area. Large number of applications are coming based on mass spectrometer, but the fundamental concepts of mass specs remain same. You are looking at a technique to identify protein, analyze protein by producing the charged molecular species in the vacuum, and how well you can separate them with the magnetic electric field based on the m by z ratio or mass to charge ratio. So any type of, I'm sure you would have heard some mass spectrometer in your institute or nearby, or you may already have it one in lab, like Q-TOF mass spectrometer, multi-TOF mass spectrometer. You must have heard new Orbitrap mass specs are coming. So all of these instruments are having certain, unique features from the instrumentation point of view, but conceptually, they all will be performing in this kind of manner. So let's say you want to do proteomics using mass spectrometer. First, you have extracted the proteome, the proteins from the complex samples, which you have done digestion with the enzyme like trypsin to make them cleave them into peptide forms. Then you want to fractionate those peptides, complex peptide, because let's say we had thousands of protein here and now we got a couple of 100,000 peptides. Now you want to fractionate those peptides using chromatography. And then they are coming to the mass spec. Mass spec has these three major parts. One is the ionization source, second is the mass analyzer, and third is detector. So these peptides, which are in solution form, they are coming to ESI or the electrospray ionization, one type of ionization source, where they get ionized in the gaseous forms, and then they move inside the mass analyzers. So any mass spec which you would have seen will have either ESI or the multi-ionization source. And inside part is the different type of mass analyzers where we can have quadrupoles, which is I'm shown here. You can have the time of flight as well. You can have the Q-TOF like quadrupole and time of flight. You can have quadrupole and orbit trap. You can have ion trap, and so many type of mass analyzers you can have. Again, I'm not going into all of those possibilities, but uh, there are many type of mass analyzers available. Idea is, can we make use of two different type of mass analyzers, so that we can take two different properties of those mass analyzers, right? For example, if you take quadrupole and time of flight, so Q and TOF together. So it means the properties of quadrupole, how to separate the ions in the revolving electrical field, and also time of flight, how much time they take to fly inside the tube. So you're taking two different properties to separate those. Now, in any of these, this is known as tandem mass spec if we are using two different type of mass analyzers. And in between these two type of mass analyzers, in this case, what I'm showing you is the quadrupole only, quadrupole Q1, four set of metallic rods to separate the complex ions. And this is the Q3, which is again another set of quadrupole rods to separate the ions. In between these two, there is a collision cell. The collision cell aim is to, for example, from here, when we have too many ions coming in, it selects most intense fragment, most intense parent ion, which is getting further fragmented in the collision cell. And now you are generating MSMS fragmentation from it. So what is happening, if you had only used one type of mass analyzer, you are going to have the PMF type of mass peptide, mass fingerprint, or only mass of that particular thing you can generate. That is known as MS. If you were to do the fragmentation, MSMS, now you can look at the B ion, Y ion, and you can use that information to derive peptide sequence. So this is a very complex slide, but one slide has so much information. First of all, a workflow of proteomics with mass spec involves protein extraction, protein digestion, complex protein fractionation, peptide fractionation, followed by moving to the mass spec. First is the ionization source separation, followed by mass analyzer based separation. Then those masses reaches to the detector to show you the peptide, the spectra. In between these, there are concepts of MS or MSMS or tandem MS. When we are using two different mass analyzers, idea is to fragment those ions, take the most intense precursors first, do the collision cell with the inert gases to fragment those ions, and generate further mass fingerprint from that, and then generate MSMS ions. So in this way, can do many applications, different type of application depending on your requirement. For proteomics, we always do MSMS because we want to know the peptide sequences, and then from those data, we are going to search the databases to know these peptides belong to which protein, right? And then from there, we will know that, okay, this many peptides are coming from human, and some maybe even, you may have contaminants and all as well. to be very confident about that you're the protein which you are identifying. Again, just to keep it very clear, we are not doing end terminal sequencing. We are not sequencing the entire protein here. We are only looking at few peptides of some proteins, right, of every individual. So now your database search and your filtering criteria becomes quite important, because you need to make sure that you are searching with very high stringency criteria. At least the protein should have two unique peptides. If you are searching, let's say, based on one unique peptide, that one unique peptide may be common to human and monkey. But if you're looking at least more than two unique peptides, that is very good probability that it is coming only from human. And you need to increase the length of the peptide for search. For example, if the peptide was only 6 amino acid long, it may be, can bind to other or have similar regions in the other organism as well. But if you're looking at 15 amino acid base long peptide sequence, then that will be pretty unique to that organism. The two unique peptides, non-nested from different regions of the protein, and then using that information to search for the at least 15 amino acid long peptides with very high filtering criteria, 1% false discovery rate, all of that being used to search the databases. One of the very latest instrumentation in the field, which we also have in our lab, is Orbitrap Fusion Mass Spectrometer. And here, again, any mass spectrometer will have common, the instrument parts, but the middle part is the most creative, where different type of mass analyzers are being used. The first part is ionization source, as I mentioned to you, ESI, electrospinization. All your peptides are from solution form are coming here, and now you're moving them with the very high voltage and temperature and the voltage differences. You're trying to generate the ions, and those ions are going to move inside the mass analyzers. In this case, fusion is tribrid mass spectrometer, which is having three different type of mass analyzers. It is having quadrupoles, orbitraps, and ion traps. So, based on these properties, one could utilize this instrument for different applications. So, therefore, these ions are getting queued up here in C trap coming from quadrupole, and you can send them to orbitrap for MSN fragmentation. You can send them to ion trap for further MSN analysis. You can use different type of fragmentation patterns here for HCD or ETD. You can use them for the peptide sequencing, or even looking for the PTM type of analysis. A lot of things can be done on these type of mass spectrometer, but it's not the only mass spectrometer. As I said, the basic concept remains same for all of these mass specs like this. Like here, if you now dissect out what I just showed you, this is the ionization source, the one part, and the mass analyzer what I showed you for fusion, having three different type of mass analyzers: quadrupole, orbitrap, as well as the ion trap. So that will keep changing depending on the instrument type. Now, based on some of these instrumentation, now we have better capability of doing quantitative proteomics. With the gel-based proteomics, I have shown you the 2D dice technology, which can look at every spot which is having PI value and molecular weight. And then you are going to compare them from control C1 to T1 and C2 T2, C3 T3. And then you will look for the most consistent and significant changes, right? But the gel can only resolve few hundred protein, not at the scale of thousands of protein, what we are looking at in this kind of quantitative proteomics approach. So therefore, various type of mass spectrometry based quantitative proteomic technologies have emerged, which means you are not going to separate protein on the gel. Directly you extract the protein, digest the peptides in the first technique which I'm talking, tandem mass tag, TMT, label them with these isobaric tags of TMT. Uh, Isobaric means you're not going to change any different mass on the all of these six conditions which you want to compare: three control and three treatment. Everything gets some sort of reporter ions, but overall they're not going to change the mass of the overall proteins. Then you are going to do the LCM SMS analysis, and now six conditions have been labeled. You are going to look at what happens to my every peptide in all my six conditions, and look at their trend and the patterns and the fold change information, and that in that way you can now see which proteins are showing significant change. What is the major advantage here is that every protein for which you are looking at the fold change or differential analysis, you have already acquired the MSMS spectra. So you can at least know the name of protein right away. In case of gels, once you see that visually the gel is looking good, that spot is looking very different, and now you have to cut that spot and do the in-gel digestion of that spot, and then you go for mass spec analysis. And many times spots are having very less amount of peptide. So you will have the miss of the that identification. You can identify that protein because the amount of protein available in that region was very small on that spot. Here, everything whatever you are able to do, you are already generating the MSMS spectra, so 100% identification is available of thousands of protein, right? So that's pretty powerful. This is quantitative approach using tags, tandem mass tag. This is known as label-based quantitative proteomics of mass spec. Now, label-free is idea is we are not labeling any tag or any kind of weight on the proteins, but what we are looking at running every sample individually and then trying to measure are there some spectral difference in the number of spectra which we are acquiring on each detention time? And if that is the case, then that can be utilized for comparing my control versus treatment. So here the idea, the analysis becomes very powerful. You are not labeling any condition. You are running every sample individually, not unlike here when every sample has been pooled. Here every sample has to be done separately. And you are going to compare that what is the spectral count of each protein and peptides across my different conditions. However, the one major problem which we find in the label-free quantification based quantitative proteomics is a lot of missing values are seen when we are comparing control with treatment. You will see a lot of samples don't show any data point from the mass spec. So therefore, those are known as missing values, and that is one of the major challenge with the label-free data. So to further overcome some of these methods, another technology which is coming forward or methodology is coming forward is known as DIA, data independent acquisition. These two are known as data dependent acquisition broadly because you are acquiring data from a specific type of precursors, you are fragmenting those precursors and generating data from it. Here, what happens in DIA, we are generating some very high resolution libraries. So, let's say from my given all the samples which we want to analyze, I'll make some pool of it and first run that pool of sample for a specific M by Z region. So, let's say I'll run my sample for 400 to 600 M by Z. Same sample I'll run for 600 to 800 M by Z, 800 to 1000, 1000 to 1200, like that, right? So in that way, for each of those small M by Z region, we are generating very high resolution libraries. And then I'm going to use that data to build my library. Further, I'm going to run my sample like earlier, individually, every sample. But now once I will analyze my data, I will overlay my data with this library, which is very high resolution library. So now I have higher probability and chances of analyzing that data with no missing value. So this is working very well, and that is currently the whole field of proteomics is moving towards data independent acquisition. This slide is slightly complex, especially some of you are beginners in the field of proteomics. Something are not clear, please do feel free to ask me. You can always even ask my opinion if you are going to start this kind of experiments or project in your lab, that what can be the best methodology which can suit your requirements, and we'll be happy to talk to you even here or some other place or by email. So other field which is emerging is known as targeted proteomics. So for example, we have identified some specific targets after comparing our control and treatment condition, and now we want to validate them. I'm sure for validation you have used some antibody-based methods. You have used Western blots. You have used ELISA. You have used some of the, immunohistochemistry. You have used different type of fluorescence-based methods. However, what we are doing in the proteomics, like all of these experiments, what we are identifying is the peptides, peptide sequences. So can we now, since now we have information for those important peptides which are showing differential response from control and treatment. So once I now know that these peptide sequences, why not I validate that only rather than raising antibody? So now we have come up and people in the proteomics field has come up with a new ways of validating the hits known as target proteomics. One of them is known as MRM, multiple reaction monitoring. Other is known as PRM or parallel reaction monitoring. In MRM, that is based on the triple quadrupole mass spectrometer. Let's say you already know which peptides which you want to validate across hundreds of samples. So you're only telling software that this is a peptide sequence I'm interested in, and with the hardware connection, it is only going to pick up those peptides. Further, you're going to fragment those in collision cell, and then you're going to generate the MSMS of that, which will make you the time versus ion intensity graph. Alternatively, and this is what how it looks actually in reality, the data looks like these. These are known as transitions. Data analysis being done on software known as Skyline. Additionally, we also use PRM or parallel reaction monitoring, where A quadrupole is connected with the orbit trap. In between, of course, we have collision cell, and ideally you are generating peptides and validating all of them parallel in this approach. So these are the two main methods of doing validation as well. Let me take you to my lab and show you at least how these instruments look like, what kind of thing they can be, they will be doing. I've already talked to you a little bit more complex part of concepts of PRM versus MRM, concepts of DDA versus DIA, concept of the TMT tandem mass tag with label free quantification and with the DIA data input acquisition. I think you need some break now, so you can watch this clip from my lab, which I have recorded for the facility. My lab is primarily focusing on proteomics-based applications to investigate different types of clinical problems. As you can hear a lot of noise here, I'm trying to show you some of the technology which we use in our lab for doing the clinical proteomics research. Instrument, what we are standing nearby is one of the next generation sequences. And I'll explain you shortly that when this SARS-CoV-2 virus started showing its impact and COVID-19 was supposed to become a pandemic, we first needed to know about the sequence of this virus. And that's where the next generation sequencer and these kind of technologies are very helpful to decode the genome and tell us that what is the genomic composition of a given organism which can be utilized to further detect that type of organism. So, we thought can we detect these viral peptides and whatever responses are happening because of the virus in human, in host response, using mass spectrometry technology. So first of all, we try to look for what all proteins are present in the virus, which is using these mass spectrometry. And then the goal was to look at also the most proteins which are getting modulated in response to this virus. So this instrument is known as LC-MS. The first part is the electron chromatography or the nanometry, and then we have electrospray ionization, and then we have private mass spectrometer, which is one of the orbit trap technology coupled with quadrupole and ion trap. So here the goal is to have this very high resolution mass spectrometer can be tried to separate the complex protein mixture, and that comes under deep proteome investigation. So we first looked at variety of samples from the patients from the nasopharyngeal swab, which is currently being used for the RT-PCR detection, as well as the plasma sample, which gives the good pathophysiological information from the patients. And these samples were deep profiled in short-term proteomics using the LC-MS investigation. So after the deep protein profiling is done, the next goal is can we develop some of the simple assays or tests using mass spectrometers, which can be utilized for two activities. One for looking at the SARS-CoV-2 viral peptides, that is in some way an alternative to the viral peptide detection. Or second, it could be can we look into the host response and the protein, which could be prognostic biomarkers for disease severity progression. So this instrument is a wonderful mass spectrometer, which can be utilized to specifically look at the peptides which are showing maximum change in response to given disease. Or for that matter, if we are looking at the infection of the virus, we are only looking at the specific viral peptide and totally ignoring all the good protein types. So this technology works in the field of proteomics, known as targeted proteomics, where we develop this kind of assays using selected reaction monitoring. And our goal over there is to look at the specific transition of these peptides, which can be then monitored further to detect whether the virus is present or not. Or if we are looking at the host response, then we want to know whether a specific peptide from the host side is showing any alteration or the change in abundance in response to the severe progression. So given that whenever a viral induces a host immune response, several metabolites also get altered. And we thought can we investigate the plasma samples of
COVID-19 infected patients and try to investigate which are the metabolites which are getting further in response to COVID-19. And again, we use another type of master spectrometer. This is one of the only tools which is master spectrometer. And along with it, we have this UHPLC. So here you can have much larger volume of the sample to be injected. And that samples we are looking for with very simple organic solvents like methanol extractions. We want to look at which are the metabolites which are coming from the patients, and these kinds of feeds will simply detectors that can we start looking at the regions where the metabolites are maximally getting changed, and that of course requires one of the condition analysis to find out which of the metabolites which are getting changed. Okay, so let me now take you to our actual molecular biology lab. And of course, I have to wear the mask now because of the COVID situation. So we will just give you brief, virtual tour of the lab where people do sample preparation and all kind of molecular biology based work. Of course, you're going to look at a variety of fancy instruments, but something as simple as this thermal cycler, that itself also is very crucial. And at the time of pandemic, that really served its purpose for looking at how viral amplification can be detected from the mRNA using real-time PCR. So again, some of the basic technology which we talk in the course are routinely used in the molecular biology labs, and that have really helped during this pandemic time. I'll take you to another instrument which is quite helpful. And let me now show you another technology which is microarrays, and we are talking about protein microarrays here. So these are high density array which is having thousands of protein printed on the small microscopic glass slide, and this actually contains almost 40,000 features consisting of 18,000 plus proteins. So now think about on these kind of high density chips with very small volume of patient samples, you can screen different type of biomarkers looking for antibodies or various type of protein interactors, right? So that's where I think you can think about with modern technologies how looking at interaction and interactomics have become quite feasible. So on the screen, what I'm showing you here is one of the slides showing the protein expression with the GST tag, and that shows that we have to do a lot of placing the grid, etc., to make sure that we can find out the right intensities of these proteins, and then that could be utilized for subsequent big data analysis. This is a small scanner which is quite helpful for scanning these chips and different wavelengths. Using one of the instruments which is not so commonly used in proteomics, but especially for the COVID investigation, we look at FTIR based detection. Can we look at simplified methods to detect virus very quickly? And that's where FTIR instrument was quite helpful. Of course, for many of the things which we talk in the proteomics or genomics or a big data field, you need to prepare thousands and thousands of samples, right? And that's where having this kind of liquid handler becomes very handy, so that you can prepare samples in 96 well plate format, and you can prepare them quite in the robotic manner, and you don't have to rely on manual way of handling the pipettes. So again, some of these things can be helpful if you're looking at proteome-wide or genome-wide investigation. So in the clinical proteomics or clinical omics field, you are heavily relying on large number of clinical samples, right? And that's where the sample storage becomes quite challenging. I'm sure while discussing about COVID, you have heard about the storage issued of different vaccines, especially the RNA-based vaccines, which is Moderna and Pfizer they made, and storing them at minus 80 degrees. Those kind of temperature can be very challenging. Now, what I'm standing here is in front of one of the freezer, which is having minus 86-degree temperature, and all of our clinical samples and different type of clones which are required for doing these kind of research are actually stored in that kind of condition. So you can imagine that to really prevent any kind of degradation of the samples and any contamination, we want to really store them to that level of freezing condition, right? And that's where doing this kind of research becomes quite challenging and resource intensive as well.
And finally, let me show you that this is one of the big data processing server where you can now take huge amount of data sets which we acquire from these instruments of mass spectrometers or NGS or PCR, and then we try to investigate that how to combine the data sets and look at it in a much more holistic manner. And that's where you need much more computing power. And of course, many of you who are from the engineering background and who are more intended for the data analysis part, I think you have to think about how to best utilize the computing power to process the big data in more meaningful manner. All right, so now you have seen the lab and you have seen the instruments. While the instrument looks very fancy and everybody feels that okay, learning them is the best thing to do in the proteomics, but as I mentioned to you, the sample preparation is the most important part. If you bring in good sample, these instruments are very capable of generating the high quality data, and eventually you can then do a good data analysis to identify those proteins. Now, all of our efforts of proteomics is actually following the maybe a decade long delay from genomics. So all the human genome project got completed 2002, whereas the first draft of the human proteome map was accomplished in 2014. And on the left side, as you see, these are the mass spectrometry based approaches to do the draft human proteome map. And on the right hand side, it is a tissue based proteome map. Again, or both the approaches try to look for all the proteins of human, but they could reach only 70% of the human proteome map. Very recently, with the effort of Human Proteome Organization, many research groups, including ours from IIT Bombay, now we have come up with the 91% draft human proteome map, which means now we have at least 91% protein of human for which we have verified data from the instruments. Still, there is a quite a bit gap, which is known as missing proteome, or 9% proteins are missing, for which still there is a race is on to identify those proteins and characterize them better. Just to summarize my first part, I try to give you some idea for proteomic technology, especially mass spectrometers, which is complex, but I think it's very important right now in the growing field of proteomics. Understanding some of the mass spectrometry configurations are very important. Of course, I did not have sufficient time to talk to you about all the, the physics behind each one of those configurations. But if you have more interest, please do follow some of our videos where I have talked more about each of those type of technology as well. But what is really important that many type of hybrid mass spectrometers like QTOF and multi-TOF, plus looking at other tribrid mass spectrum like orbitrap fusion, they're currently being used and they are actually enhancing our knowledge of proteome-wide screening. Let's now change the gear and briefly talk to you about some of the non-mass spectrometry-based protein technology, especially protein microarray-based platforms. So while mass spec was able to give us in a very unbiased way all proteins which are present in given sample. In case of microarrays, you need to know the contents or the proteins beforehand, what you want to analyze, and you need to print them on a miniaturized microscopic glass slide which you have used, or nitrocellulose membrane or gold slide, something of that size, which is microscopic glass slides, what we use for cell biology. On that slide, different type of things can be printed, and those are known as different type of protein microarrays. So I'll just enlarge each one of those side by side, one by one. For example, the first one, what you see, antibodies are printed on the slide, and that is no. If you are putting your samples labeled with Psi3 or Psi5, that is known as direct labeling based protein microarray for the antibodies. Let's say now you have again printed antibody and you have a sample. That sample you are probing with the secondary antibody, which is conjugated with the fluorophore. Now that is a sandwich based assay you are doing. Or you can print a protein, you can print the tissue or the tissue lysate on the array and then probe them with a specific antibody. And now you're looking at the abundance of that given antibody. So that is known as reverse phase arrays, quite a bit used by the clinicians in their clinical hospitals. All these three are based on antibody-based approach. It means we are using some ways of antibodies to analyze our proteins, and therefore these are known as abundance-based protein microarray. What we are looking at, abundance of a specific protein based on antibody interaction, anti-antibody interaction. Additionally, one could also look for without antibodies, what is the function of the proteins. Most ideal arrays will be if you purify those 20,000 proteins and print them on the chip. Now you got a human proteome array which is having all human proteins. But purifying that many proteins are not easy, which I'm sure you will appreciate that purifying even one protein can be cumbersome, and purifying 20,000 proteins can be quite cumbersome. But there are some group who have done in the world, but it's not easy for everybody to do that. So many times when in the mass spec is giving us peptide sequences, can we just synthesize those peptides, and those can be done with the very easily one could do with the different type of chemical synthesis of peptides. And those peptides could be tagged with different type of tag like histidine or GST, and then use them on the array. Or could we also use the cDNA or the PCR products and add the in vitro transcription translation mix to make the protein on the chip itself? These are known as cell-free expression-based array. One is known as nucleic acid programmed protein array or the NAPA. Other is known as multiple spotting technique. So again, what you now at least can take home here, that there are different type of protein microarray platforms available. And again, depending on your application, you can choose like mass spectrometry technologies, even different type of protein micro technologies are available, right? So just one as a one example, we of course we have done on the human proteome arrays as well. All the human protein we analyze for different biomarkers. But I'm showing you here another type of array, which is peptide microarray. So in the time of COVID-19, in one of the German collaboration with the paper print array, we got these peptide arrays where all the protein of SARS-CoV-2 virus and every proteins, all the sequence they took, then different 15 amino acid long peptides were synthesized, and these peptides were in the tiling format. For example, for a spike protein, every sequence with the overlapping fragment so that you can represent the entire proteome on the chip. So that was being printed. And now, after printing those arrays, one could take the patient samples and try to probe those samples to look for where they're binding with the virus and what is the most immunogenic region to look for humoral immunity, to look for the potential vaccine candidates. So that kind of experiment can be done. Let me show you one clip of microarray workflow to get you some idea about how microarray peptide microarray experiment can be performed in the lab.
Today, we will be demonstrating peptide microarray technology. The first step involves reagent preparation, namely washing buffer, staining buffer, and Rockland blocking buffer. After preparing the reagent, we move on to incubation tray assembly. Unscrew the incubation tray by slowly turning the screws diagonally. We will be using two blank glass slides as balancers, since today we will be using only one microarray chip. This is the pepperprint microarray chip. Slowly take out the chip using plastic forceps and place it on the incubation tray. Align the chips properly onto the tray. Now, allow the gasket to be placed over the slides. Remember to keep the glossy side of the gasket on the downward side. Align the silicone gasket over the slide and tightly seal the incubation chamber using the screw. Remember, tighten the screws only diagonally. This will ensure that the slides do not break. Next, add 2 ml of staining buffer onto the chips. This step is called equilibration of peptide microarray chip. Please note that we are not touching the microarray chip at any point. Now allow the equilibration process to happen for 15 minutes. aspirate and remove the buffer and add 2 ml of blocking buffer. Slowly add the buffer onto the slides. Allow for 30 minutes of incubation. The next step is sample incubation. To the staining buffer, we will add patient plasma at a dilution of 1 is to 200. This is the patient plasma. Now slowly overlay the diluted plasma over the slide and incubate overnight. This will be done in a cold room at 4 degrees Celsius at 140 RPM on an orbital shaker. On the next day, remove the incubation tray from 4 degrees Celsius and aspirate the solution. Now add washing buffer thrice onto the chip. This step is done secondary antibody. Prepare of dilution of secondary antibody 1 is to 2000 and then slowly overlay the secondary antibody on the microarray chip. For 45 minutes, allow the incubation to happen at 140 RPM at room temperature. Make sure that you do not expose the slides to light at any given moment. Now remove the buffer and wash the slide thrice using washing buffer. Now gently take out the slide and put it in dipping buffer. This is done to wash away all the salts that may be over the peptide chip. Use a pressurized stream of nitrogen to completely dry the slides. Finally, we will scan the slide. This is the microarray scanner, GenePix 4000B. We are using manufacturer recommended settings for scanning the slide.
All right, so now you have seen the peptide microarray based workflow as well. Very briefly, I'll talk about one more technology, which is label-free biosensors. So now, since the microarray, they involved some sort of fluorophores for signals, can we analyze the similar type of high-density approaches, but without having the fluorophore? So that's where surface plasmon resonance, SPR or SPR imaging type of technologies are very important. What we are looking at here, like let's say we have a prism, a light source, we have a gold slide, and if you are placing your, let's say, antibody on the gold slide and proteins are flowing, as soon as proteins are binding to the antibody, there will be change in the refractive index of the medium, and that we are monitoring in terms of sensorgram. What you're looking at initially, when there's no binding, it is the baseline static. As soon as this binding happens between the antigen antibody, now you see some sort of binding get started, which is on rate. And after you have binding is completed, you have floated and saturated that, then you are flowing more buffer and that get dissociated. Now you see the dissociation curve or the K small d. So on rate K small a, on rate K small d. And since you are generating some sort of numerical values for both Ka and Kd, now you can generate the kinetic data. You can look for the K capital D, which is dissociation constant. So in the microarray approach, we were looking at a binding is happening, yes or no, with the signal. In this case, we are looking at binding is happening, but also what is the strength of binding, what is the on rate and the off rate. A newer approach, so again, like SPR is very useful not only for proteomics but also for auto biopharma. They look for different type of drug characterization. But it becomes very important that how important that binding on rate and off rate is. Think about the context of paracetamol, for example. If there's a pain, you want to get it very fast relief, right? So the binding has to be very quick, so on rate. Whereas if somebody is taking a sleeping pill, one would like to use that to work for a long time, and that's where you'd like to has it released very slowly. So again, it's not only about a drug is binding to the targets or receptors, but also what should be more on rate, what should be more off rate, and that is being controlled with these kind of experiments, and one could measure them. That's very important for the various type of protein drug interactions, but also for biology, a lot of protein biomolecule interactions are being measured using label free proteomic technologies. Due to the time shortage, I'll not talk about many other approaches which are coming forward. But while I was doing my postdoc at Harvard, I was also involved in developing some new technology platforms of how to integrate protein microarrays, high density arrays with surface plasmon resonance imaging based platforms so that we can make the best use of the microarray along with the imaging of the SPR so that we can do high density but without labeling the proteins with the fluorescent dyes. And likewise, there are some other approaches which is coming forward, like nanowire sensor arrays, carbon nanotubes, nanohole arrays, spectral reflectance imaging biosensor, planar wave guide arrays. You can read this article, and also there are other review articles which will give you some idea. Let me show you one SPR experiment slightly briefly from our lab.
Sensor chips, biosensor chips, on which we immobilize one of the interacting partner and we pass the other partner. The partner which we immobilize is called as ligand and the other which we pass is called as analyte. So in DRPO TP100, there is a chip compartment where we dock the chip. Okay, you put your chip here, and this is sample compartment. You will put your sample here, and this is buffer compartment, and this is discard. So you immobilize your one of the interacting partner, and then we will pass the
लाओ
other partner, and
रिमोट
you will
लाओ.
get the this association and the dissociation.
ओए रिमोट देख उधर है.
So for
पूरे दिन से टीवी चल
immobilization,
रहा है.
you use different two types of
बंद
strategies.
करो.
One is
पूरे दिन से
capture
टीवी
and
चल रहा है
other
इधर.
is direct. Either you can directly immobilize one of the interacting of the ligand or you capture. For example, in direct immobilization, you use different types of chemistries like amine coupling, thiol coupling, or you do aldehyde.
तुम्हें देखो
So
पापा
for
आएंगे.
example, in amine coupling, your amine group of your protein is involved.
देखो देख के
So
आओ.
this chip has a carboxy mechanism. You activate that group and you this activated carboxy methylated group will bind to the free amine of your protein and your protein will get directly immobilized. For example, in capture method, you have nickel NTA on your chip, for example. Also, nickel NTA will, so this step protein will get captured on the nickel NTA. So this is how you immobilize your one of the interacting partner. Then you will pass the other interacting partner. Then you will get the association. Okay. After that, buffer will pass. This will give you dissociation. The ratio of this association weight constant and the dissociation weight constant will give you dissociation. Will give you equilibrium dissociation constant. That is the strength of your interaction. So after passing of your analyte, if your analyte is not getting completely dissociated, you will need to regenerate that. For that, you have to use different regeneration buffers, and you have to optimize that. So after regeneration, you will get the sensogram like this. So for example, this is the association, this is the dissociation phase, and here you regenerate. Okay. So you have different types of chips are available. On basis of what experiment you want to perform, you will choose the chip. For example, if you want to perform If you want to use, if you want to immobilize his stack protein, you will use NTA. If you want to immobilize lipid, you will use L1. If you want to immobilize step biotinylated nucleic acid, you will use SHA. This is how, on basis of your experiment, you will use different types of chips.
All right, so now you got some idea that SPR experiment. It is quite complex. Of course, you have to do it, but we are giving you at least some flavor of it or some concept of it. So proteomics is, is still very much technology oriented field, and a lot of innovations are happening, as you can appreciate, right? From gels to the mass spec to the microarrays to the SPR label free technologies. So many innovations are happening. Let me just show you a quick status of where we are in terms of the overall proteomics project for the human. So human proteome project, which is governed from the HUPO, Human Proteome Organization, is now the flagship project of proteomics activities, and there are four major pillars of that. One is mass spectrometry based, other is antibody based, third is knowledge based or the bioinformatics, and fourth is pathology based, how to link those information for the clinical translation. There are teams who are working on different each chromosome to look at which are all proteins present in every chromosome. Different countries have been assigned that task, and likewise, which diseases having what kind of protein which are getting malfunctioned or getting altered. Different biology disease HPP based projects are there. So it means BDHPP and CHPP are the two major initiatives of the Human Proteome Project, which is currently in progress. Please keep in mind some of the very important data analysis parameters. As I already referred, that you need to have two unique peptides. They should be slightly far apart from each other. They should be long. And one need to look at the good FDR values and all that. And based on some of these information, during the COVID time, we actually published this work in Nature Communication, which was a part of HUPO, Human Proteome Organization flagship project, where we have now have 91% of the human proteins identified from it, for which we have experimental evidence from these instruments, mass spec, etc. Still, there's some scope to identify the missing protein. There are many class of protein like olfactory receptors, transmembrane proteins, zinc finger proteins, keratin-associated proteins. Some of these proteins are quite difficult to, detect them in the mass specs, and therefore, still a lot of efforts are being made how to identify the missing proteome, and that's where I think is still quite a bit of opportunity there for field of proteomics. So just to summarize, I think proteomics have come quite far with different type of technologies that are being integrated, and how best now we can make use of these technology to understand biology will be the part for my next part of the next 15 minutes or so, to give you flavor of how you can utilize these technologies for understanding some biology. And of course, we do several projects in the lab, but I just chose one of them on the COVID-19. And now it will be not essentially, aim is not to tell you what all we did, but aim is for you to appreciate how proteomic technologies can be utilized in a very fast time, something like this, when we had literally no availability of anything new. Whatever you have in your instruments or regions and lab, you have to use that. And how different type of questions can be addressed using these particular technologies. So you can pay attention more toward the which platform, which methodology, what was the objective, how we did that, rather than what we did on the COVID-19. So we collaborated with many clinicians, different collaborating hospitals from Mumbai and across other places, to look at detection of the SARS-CoV-2 virus and monitoring the host response of COVID-19 using different type of proteomics approaches. I think it's past, probably history. Let's hope it will become history. And we had all seen this challenge, and the research even doing that was very challenging with the PPE kits and all. Uh, we had many objectives, which I will go one by one. I'm not going to read here right now for you, but I'll just go one by one. First was to detect whether we can. We already had the standard workflow of detecting virus from the swab samples with the real-time PCR analysis. Can we detect those viral peptides from the mass spectrometer, an alternative technology from the same sample type? And that time we had no idea whether, so if we are taking out the swab sample from human respiratory specimen, whether that will have some viral peptide, we had no idea. That was the curiosity which we wanted to address. So the same sample which clinicians were taking in Kasturba and other hospital in Mumbai, which is being used for RT-PCR to measure the CT value, some of the leftover sample we were getting it to do the different type of organic solvent based precipitation, because the first idea was to ensure that virus get denatured, right? You don't want to play with the virus. So ethanol, IPA, acetone-based prescriptions were tried, protein extraction, digestion, followed by running in the Orbitrap instrument to profile all the proteins from that given sample. But since it is human respiratory sample from the swab sample, ideally we will have a 99 point more percentage of that protein as a human only, and very very less abundant of the viral protein will be present. So now this is where the database search becomes important. What you generate here is a peptide, entire libraries, and that if you search first against the host human, you will see all the human protein. But the same data when you search against virus SARS-CoV-2 database, then you will see, okay, how many peptides are also coming from virus. And if there are some peptide coming from virus, can we now validate them using the selected reaction monitoring assays, which I talked to you in target proteomics. So that's the idea. First thing which we try to extract these, proteins and peptides, and we ran them in the mass spec. We identified, did the search with the SARS-CoV-2 database, and several peptides of virus were actually seen in this type of nasopharyngeal swab sample. That was very interesting and one good success to begin with, because now we can move forward to develop the assays. So then now we know which peptides are seen commonly from the nucleoprotein or spike protein of virus. So now we made the list of those peptides and try to develop the triple quadrupole based simple assays of mass spec. When you're doing this kind of test or assay development, you have to also have some synthetic peptide which you inject in every sample to ensure that that can be utilized for normalization and your QC quality control checks. Finally, what we developed this type of test, which was having 11 peptides of SARS-CoV-2 virus, 4 from spike protein, 3 from nucleoprotein, and 4 from replicase polyprotein, in 8 minutes long gradient. So if you take the swab sample, extract the protein from it, run in the mass spec for 8 minutes, only screening for these 11 peptides, then based on that, we are able to now say this is SARS-CoV-2 positive or negative. And since we are talking about 11 peptide-based screening, it is more powerful as compared to when you're looking at RT-PCR based analysis, because that is based on only 2 genes. Also, since you're all coming from technical background, you know that extracting RNA can be tedious. It is very labile. Storage is problem, and then you are doing the amplification to uh, cDNA, and you are looking at the CT values eventually from that. So therefore, here we are looking at whatever peptide is present. That's all. You're not amplifying anything. You're not changing anything. And many times when we had seeing the variant change or mutations happening, peptides are more powerful because there is no miss like RT-PCR in that way. So this was one of the patent which we filed. We have also publication from this, a methodology which we published in Star Protocol of Cell Press, where people can now go and look for those detailed protocol and try to, follow that experiment. So this is quite interesting, good beginning of doing SARS-CoV-2 research, despite in the challenging time. But we realized the major challenge was the instrument cost. If we really wanted to reach out to many path labs and places, they did not have the instruments and the specialized manpower. So that was one challenge, but at least the kind of test using proteomics technologies could be developed. We also then thought, since we are looking at nasopharyngeal swab samples, which is having human proteins, okay, let's look at what kind of human protein response are getting changed or the host response getting changed. But throughout, when you are doing hundreds of sample analysis, every day when you run sample in mass spec, each sample, let's say, takes 2 hours time. Then you run 2 hours of blank. So it means in 4 hours you're only running one sample. You have to put some time for quality control checks, and also hardly 5 to 6 samples can be run in a given day on mass spec. And if you are going to run 200 sample, it will take a couple of weeks time to do the analysis. So then you need to have some sample which is the same sample which you should be running every day or running after every 5 samples. That is known as the QC pool sample. And that becomes very important at the end of experiment to compare across from the beginning till end when you were running that project. When you are now comparing only pool sample, how uniform the pools are. That was very important for us to show to the reviewers that pool qualities are pretty good. And based on this type of host response, now what we are able to segregate, this is the true negative in the blue color, red ones are positive, and in between is the category which is the violet colored category, which is uh, true recovered category. That was very interesting because these individuals, they are although having the CT value negative, but they were still carrying some viral peptides, and therefore now they are actually grouped under the positive groups. This is something which clinicians got very excited about, and felt that mass spec can do some interesting things. At that time, if you recall, clinicians were measuring, interleukin 6, lactate dehydrogenase, aspartate transaminase, C-reactive protein, some of these proteins to indirectly help them to see, like, which individuals are more affected with the COVID-19. And with the mass spec, with the same sample, now you can look at all of them protein same time. You don't have to do separate test, right? And in the biochemistry lab, every protein you have to test separately with different type of assays and fluorophores. In this case here, we are looking at everything in the MRM-based assay. We are only giving the peptide sequences, and all the proteins are in present given sample type. So one could screen them all together. This was very interesting for the clinicians to convince that we can now come up with a new test which will be more reliable for them. So now next, which we wanted to look at, individuals who are suffering from COVID-19, can we take their plasma samples? And we had already done some work, similar work in the malaria, which was published in Nature Communication Biology just recently before the COVID started. So fortunately, we had all the tests and reagents available for doing that kind of work. So almost 200 individuals were recruited, which were COVID positive or negative. And from each of those plasma sample, we did depletion to remove the abundant proteins, ran them in the mass spectrometers, and then did all type of bioinformatic analysis to find out are there differences among the mild type of COVID-19, non-severe versus severe type of COVID-19. And some interesting proteins showed that there is a differences between non-severe and severe form. And finally, based on these large number of protein lists, we did machine learning to narrow down which proteins are most influential. And interestingly, apolipoB protein, tryptophan gamma chain protein, some of these turned out to be quite interesting protein. So we thought now we can use these proteins to validate between non-severe and severe form using our multiple regression monitoring based assays. So now what we are looking at comparing non-severe and severe form and angiotensinogen, apolipoB, tryptophan gamma chain, and serpin G1 proteins. So now 4 proteins turned out to be very important to develop this test, MRM test for the COVID-19 prognosis or transition from the mild versus severe form. And that you can see the levels were very down in the non-severe, were very high in the severe form. Additionally, we also understood a lot of biology of it. And so again, this test was also patented and very, got a lot of attention from various stakeholders. We also understood the biology about what is happening with the virus and host interplay. Some of the pathways, like leukocyte activation pathways and complement pathways, were quite perturbed. We then further, based on the reviewers' comments, we took around 25 individuals for which we collected longitudinal samples, because what the previous study was different mild individuals, different severe individuals, but now we are looking at same individual, different time points, moving from the mild to the severe form, which was quite challenging to get samples like that, but still we got quite a few to look at both metabolomics and proteomics, and came up with this longitudinal study, which was in JPR cover page. Additionally, how fast we can do this type of test. Now we know there are changes happening in the blood, which can differentiate the non-severe or the severe form. So then we developed this test on the FTIR, a very simplified test, which along with our collaborators from Australia, we developed the models, which was almost 94.1% sensitive. And based on this, now in a very few minutes, one could differentiate these type of severe form, which was quite a bit taken by the media, and it was published in Analytical Chemistry. So this was a rapid test for COVID-19 severity detection. Uh, I have already talked to you about the overview of this work of plasma. Uh, we also compared first wave and second wave of COVID-19, the Delta wave, and we found that in the second Delta wave, some of the antimicrobial peptides were quite more prominent, which led us to think about the effect of various type of co-infections which were part of it. We have also done more recently about taking some of these cues in mind. We have done a lot of metaproteome analysis to identify, in addition to human protein and virus protein, which other pathogenic interactions are happening. And that has yielded some very interesting output, which I'm not talking right now, but that is a separate publication we already have on that as well. Some clinicians from Jaslok Hospital in Mumbai, they were keen into they were working on the reproductive biology to look at if the individual men are suffering from COVID-19, they have recovered, are their fertility parameters are fine. And so they looked at the COVID-19 recovered individuals 20 and the healthy men, and their semen sample, the proteomics of that was performed. And what was interesting to see that some of the key proteins which are linked to the motility, fertilization, and spermatogenesis, like seminogen protein 1, autodefensin protein, and pro-saposin protein, though were very still very down-regulated. It means their levels were much lower as compared to the basal level of the proteins. We did validation using the triple quadrupole based MRM assay, which you can see here, and that actually led us to think that these malware still not all the proteins have recovered to the normal level. They will take some more time, and again that was quite a bit picked by a lot of media. So it means one need to look for the impact of COVID-19 to the long term, and long COVID. You have seen a lot of studies that are happening right now. Given that viruses keep changing its nature, different mutant forms, I already showed you the experiment of peptide microarrays, how to screen these kind of immunogenic regions. So based on that, now we have the chips where we have both wild type and mutant forms, and we are screening against large number of patients to look at what is the pattern of every peptide, every protein across different individuals who are having different type of severity, also the different mutation forms, where these peptides are located in the 3D structure, and can some of these targets be utilized as a vaccine candidate. So just to summarize this part, we developed the MRM based mass spectrometry based test for detecting virus. We have come up with the the prognostic assay for plasma. We have identified some novel pathways. We have looked at the long-term impact of the COVID on the human reproduction. uh, we were kind of one of the first groups in India to lead some of these mass spectrometry based proteomics approaches for COVID-19, and some of these tests were developed. I was also invited by the Nature to give a webinar on how proteomic technologies are being used for the COVID-19 research, and that was kind of very important recognition to have that webinar for global world. So, just in last few minutes, I'll give you one more recent work which we have done on the human brain. We have been working on different type of brain tumors, and what we realized that identifying proteins from the even normal brain is not possible, not easy, because doing the brain research is challenging. So when we were working on different type of brain tumor like glioblastomas and meningiomas and other tumor type, comparing the normal level itself is not available. So we collaborated with one group from Spain as a part of the Human Brain Proteome project of UPO, and we did several normal individuals of the Spain and India. We looked at their region and sub-regions to profile all the proteins from those and the phosphoproteins from each regions, and we made this database brain plot. Then we also added now more data sets from the various type of other databases and for different diseases. So currently the brain plot, which is already available publicly, is one of the most comprehensive database for human brain disease. It has 6 domains: interhemispheric brain proteome map to look for all the proteins of different regions, subregions; human brain disease atlas; brain disease transcriptome map; brain disease proteome map; brain disease drug finder, as well as brain disease marker curator. I'll just walk you through quickly. So if you are looking at any brain diseases, you can first go to HBDA domain, human brain disease atlas, look for which proteins are which disease you are interested in. From there, you are looking at, let's say, specific markers of a given disease. So in this case, let's say we are looking at meningioma. You click on that, and different type of rankings are given by different databases. But we have also come up with our own ranking, but because we have analyzed the data ourselves. So then now you find NF2 as one of the key protein coming out in the first rank, which is also a known marker of the meningioma. So likewise, you can go for any disease and find out what are the best markers available. You can also look at the trend of the various proteins across different type of tumors and different type of diseases of neurodegenerative disorders, and also the transcript profile of those. Here I have shown the Parkinson disease. Uh, brain drug, brain disease drug finder allows you to look for your proteins of interest. What are the current clinical trial available, and what is the status of those happening? So that one could analyze. And for example, if you want to study the glioma, you first go to Human Brain Disease Atlas. You look for the best marker and look at the ranking. So TPP53, P10, IDH, EGFR, MGMT. These are the top rank coming in our data, which is also known markers of the brain tumors or the gliomas. You can look at their profile at a transcript level, and the again which of the drugs are being used for the currently drug trials. And likewise, for various type of neurodegenerative disorder, you can work on any of these diseases and look at what are their markers. And again, we don't work on this area, but people in the field, they tell these are the very best markers like APP, ApoE, etc. for identifying these Alzheimer disease. Again, you can look at all the information at the transcript level and proteome level and what are the drug trials available. So these are the kind of community resources which we have created, and that is going to help a lot of researchers in the field to know what is the data available. And one could of course then, we are currently working on the best machine learning panels for each disease to know that which are the best markers for that given disease. That is still ongoing work. You can always visit our Proteonomics IT Bombay, a lot of video which we upload to learn more about it. Of course, given the time, only glimpse one could give you, but you can always learn a lot of things. So I'd like to thank many funding agencies which have contributed generously, a lot of companies which contribute to various center of excellence in our labs, and of course the hardworking team from both the wet lab side and computational side who have helped in contributing all of these activities. And finally, I'd like to announce you that okay, we are going to conduct some very advanced workshops at IIT Bombay for proteomics, February 17, 18, and a conference 1920. This will be one of the best activity and event which will happen in the field of proteomics. So if you're interested, you can contact us. We'll be making announcements very shortly. Thank you very much for your attention, and I'm reachable now for any questions as well as on my email ID, which is mentioned here. Thank you very much. I hope you got some idea for Proteonomics Technologies, and if you have any question, please feel free to ask me.
Thank you, Sanjay sir, Sanjeev sir. How are you?
I'm doing well, thank you.
Ah.
Is there any questions from the audience?
Participants, please ask question.
So,
Yes, please.
considering this FTIR that was used for the proteomics, so what is the sensitivity of the test? I mean, are you able to get the exact data or?
Right. So, see, when we did these plasma sample screening from mass spec gives you very accurate data. It gives you peptide sequence, which peptides are there, right? For which metabolites are there.
Okay.
FTIR does not give any of that detail. FTIR only gives you which wavelength regions are changing maximally from the your mild versus severe form. So FTIR only gives us a quick glimpse of it, right? But given that we have already established from the mass spec that these are changes happening in these peptides and these regions, now the FTIR was quick enough to pick that up just for the screening purpose because you need quick screening. FTIR is not as accurate as the mass spec. But
Yes.
FTIR model which we created with the Australian team who were expert of modeling of the FTIR data was almost 95% accurate.
Okay,
And
sir.
we used actually blinded data as well. So data was recorded from Indian samples here, and they built the model first. Then the blinded data means they did not know that what is the mild and severe form, and with 95% accuracy, it was able to detect. But I'm saying like in FTIR we are only looking at which wavelength regions are changing, but in mass spec we are knowing exactly which peptide, which proteins, which metabolites are changing. It's very accurate, but mass specs are very costly equipments, very heavy instrumentation. Installing them to the pads apps are not easy. Whereas
Yeah.
FTI is very handy equipment, you can place it anywhere, right? So that was
Yes.
the trade-off.
Okay, sir. So one more thing is regarding like the 2D and dice. So since we in dice we tag it with the dice. So if there is any novel sorry, the spot that we see, so will the die what do you call, yeah, the tag die will try to interfere with the sequence if you
Yeah,
want to.
so that's always the challenge. Whenever you put any tag, right, in any type of tagging chemistry, it
Yes.
can't be perfect. You will always have some interference. That's where told you mainly where proteomics is moving is more towards the label-free approaches, where
Okay.
you don't want to tag anything, put any tag, and directly you want to run those sample in the mass spec and look at their abundance based on the spectral count. Like for example, if control and treatment you are running individually of a given specific mass to charge ratio m by z for that retention time. If there are 5 spectra coming in control and 25 coming in treatment, it means there is a high abundance of that protein in the treated sample or disease sample.
Okay.
This is where looking at the intensity and the spectral count is now being used for the label-free proteomics. Also, when I talk to you microarray versus SPR, again you can see label-based with a label-free comparison, right? So label-based are easy to do because a lot of scanners and the instrumentations are for that. But label-free technologies are much easily one could utilize them for more accurate information. So that kind of thing you can obtain.
Okay, sir. Thank you, sir.
Any more questions? So I'll be happy if any one of you if have more query for specific projects requirement which you have would like to further use the proteomics in your own lab. We'll be very happy to help you and to give you more insight about what protocol, what methodologies are available. Also, many instruments which I showed you, uh, they are as a part of national facilities. Anybody can use it. Please do make use of them. We are very open that people should be coming and running their samples, making best use of these technologies and government resources available. And we do keep conducting the workshops, which is also helpful for training more people, and they can start gaining more confidence that they can do proteomics. So this is not just kind of a lecture to give you what we do, but rather idea was to empower you, give a better feeling of how you can use proteomics for your own research. Any more questions?
So, Namaskar, sir.
Yes.
Thank you so much for the inspiring lecture and it's very good virtual tour of the proteomic lab. So, I just have one query since I am working on the fermented food. Suppose I have some protein food and I want to check what are the different kinds of peptides that are generated during fermentation of a food. Is it possible to like find out the peptides that are generated during this fermentation process?
Right. So see, proteomic technologies are quite robust, and very generic approaches can be tried. So same way again, you have to do different type of protein extraction to get the best protein out of the sample, and that I'm sure there are available protocols. You will digest the protein to peptides, run in the mass spectrometer. You will generate lot of peptide data. Where the challenge will come for your given fruit of interest, whether there is a database available. So you need to have a query database to search against, for to identify the proteins. If that particular organism or that particular sample has not been sequenced, so you don't have that information. So that even if you have genomic database available, one could then translate them to the protein and still make use of them for the proteomic research. So database availability will be limiting factor, but doing the sample prep mass spec is not a limiting factor.
Okay, thank you.
All right, anybody else? Any questions? Any comments? Okay, if not, Dr. Sandeep, so thanks for giving the opportunity, and we'll be happy to take more questions even by email.
Thanks for giving, sir, your time. I know catching to you is also is a bit, but I always want that your lecture should be there so that people get aware of the kind of facility and work which you are
Right. So thank you very much once again.
Sir, can I give formal
we are doing
thank you?
so that thank you for yes Laksangi, please go
Okay,
ahead.
okay, sir. Yeah. Sir, we are very grateful to you because of such a motivational and informative talk that you have given. So you have beautifully explained the different concepts of proteomics, different proteomics technologies, then the protein microarray technology, surface plasmon resonance assays, and especially the applications of the proteomics technology during the COVID pandemics and your recent contributions and innovations in the field of proteomics to this subject. So I, on behalf of the UGC, HRDC, Goa University, and all the participants, I deeply express my our gratitudes to you, sir. Thank you for your time and the information. Thank you, sir.
Thank you very much, for a very nice overview and giving me this opportunity to interact with these participants and talk to you about proteomics. Thank you very much once again.
Thank you, sir. Thank you.
I'll leave now. Thanks.
Thank you.
Chalo, thank you, everybody. Good night.
Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir.
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 Thank you, sir.